In this video we will discuss about this drug loading ability of AND gate logic based system. So far we have understood that the AND gate logic based system can deliver or can efficiently target the tumors that has those biomarkers, and they have tested it with dual biomarkers and also with the single biomarkers. And for dual biomarkers they have tested for the different combinations of biomarkers, and all of them work as per the expectation with minimum non-specific findings. So now they wanted to check whether their system can deliver the drug or can deliver the drug inside the tumors or not. So for that, the drug that they have tested, they have selected is MMAE. You can check its full name from the descriptions. So once it is internalized the cells, it will trigger apoptosis. So it is very essential that their logic or their system will target only the tumor cells, preferentially towards the tumor cells than the healthy cells. So the system that they have used, that is the and the dosage capacity that they have used is similar to what has been used in this ADC, that is antibody dependent cytotoxicity method, that is 3 to 4 drugs per monoclonal antibody. So in this case they have used 3 to 4 MMAE per Aptamer or Affibody, and they also linked it with this cleavable linker, that is cathepsin-cleavable linker, so that once it is internalized it will be cleaved off by this cleavable cathepsin linker, so that this MMAE can perform its functions to trigger apoptosis. And so they have also used, apart from this MMAE, they have also used some of the commercial drugs, that is DXD, to test their system. So when they use the different combinations of this HM123 with its counterpart, that is HF1, they are able to see the cytotoxicity effects were significantly higher whenever they used HM3 compared to the HM1, that can be seen here. And when they use the different combinations of this Aptamer or Affibodies, or different combinations of the DNA splitter of different biomarkers, then they are not able to see a cell death. So this shows that the cell death occurs once the MMAE is internalized, not because of the EGFR binding or for this hybridization chain reactions. So however, they wanted to check why this cell death is high when HM3 is used, however it is not high when HM1 is used. So they move into the microscopic images, and they are able to see that when they use HM3, the fluoresceins that are formed are not smooth, however they are clustered, indicating there could be aggregations formed, whereas in the case when they use HM1, they are able to see a smooth cell membrane structure. So now since the aggregates are, and these things they have quantified also, they are where they are able to see that the intensities of this intensities where they use H2M3 is high compared to the case when they used HM1, and similar results they have seen similar results they have seen when they check the coefficient of variations. So thereafter they've seen these results for the single biomarkers. When they did check it for the dual biomarkers, there also they find the similar results, that is when they used H2M3, the fluorescence signals were in the form of segregated, and the coefficient of variation shows it is high. Coefficient of variation shows that the fluorescence intensities are significantly higher when they use H2M3 compared to the case when they don't use H2M3. And thereafter they wanted to check the internalizations, whether there is a effect of this there is a effect of this M3 on internalizations, or both of M3 or M1, both of them gets similar amount of drug internalizations. And there they for checking this they use the fluorescein biomarkers, since once drug is internalized, this SCR will also be internalized. So they decided to use fluorescein biomarkers as a good indications. So for that they first treat the samples with nuclease, so that external without permeabilizing the cells, so that the fluorescent signals present outside the cells will be degraded. After that, they check the fluorescent signals and they are able to see that when they use H2M3, the intensities were significantly higher, whereas when they use H2M1, the intensities are very low. And for the negative control, where they use Ztaq biomarkers towards this Aptamer or Affibody towards the Ztaq, which is not present on the cells, so they do not see any signals. And these things they have quantified also in the form where we can see that H2M3 internalization signals were significantly higher compared to the other two cases. This is the reason why they see more cell death, because internalizations of the drugs is high when this HM3 is used. And one of the reasons could be that the aggregations formations helps internalizations better compared to the case where there is no aggregates formations occurs. So this is how they validate their drug loading ability of their AND gate logic based system, and also the specificities by checking the controls where they see minimum or almost negligible signals. So I hope you understood their experiments and how they checked their drug loading ability of the system that they have developed. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.